Shall we start, sir? Hello. Shall we start, sir?
Yes, please.
Okay. So, good afternoon, everyone present here. First of all, I want to say sorry because our pre-decided speaker got corona positive in his family last night. Okay. So he will under isolation for at least 2 days, and he cannot join with us. Okay. So sorry for that. We will plan with the same topic with the same speaker shortly in future. Okay. So due to that, we have to change our speakers and topic as well. So we have our immediate speaker is Miss Nandita Das. So first of all, I welcome Miss Nandita Das in this webinar. So she has completed her BTech Dairy Technology from GN Patel College of Dairy Science and Food Technology, Dantewada. After that, she has completed his Masters in Technology with specialization in Dairy Chemistry from National Dairy Research Institute, Karnal. And her work was on study on milk protein profiling of indigenous breeds of cattle and buffalo. At present, she is pursuing her PhD in dairy chemistry in National Dairy Research Institute, Karnal. So I again welcome you, Ms. Nandita Das, and over to Nandita Das. Thank you.
Thank you very much, Mr. Dhruvan. A very good afternoon to one and all present here. And as Mr. Dhruvan has already introduced me, my name is Nandita Das, and today I'm going to speak on the topic that is alpha-lactalbumin and nanotubes, technology and potential applications. So this will be the flow of my presentation. That is, first of all, we'll be seeing the introduction, then the preparation of alpha-lactalbumin nanotubes, the effect of different factors on its structures, the applications of the nanotubes, and the potential advances that can be explored in future in the same regard. And at the end, I'll be winding up the whole presentation with the concluding terms. So let us see. First of all, we'll go through the introduction. So the major term in the title was the alpha-lactalbumin. So what is alpha-lactalbumin? It is the second major whey protein, and these are the you are able to see here three different structures of alpha-lactalbumin. This is the primary sequence of amino acids. This is the primary structure. This is the secondary structure of alpha-lactalbumin, and this is the crystal structure that we are able to see here. So this quaternary structure, which we are able to see here, is divided into alpha domain and beta pleated sheets. And these are the amino acid sequences which are involved in its structures. Okay, so further, there are disulfide bonds which are involved in the alpha-lactalbumin structures, and these are the amino acids which are involved in it. These are the encircled portions which are giving us the disulfide bonds in the alpha-lactalbumin, and this green encircled is the calcium binding site of the same. Now, what are the functions of alpha-lactalbumin? So beyond nutrition, the alpha-lactalbumin as a protein also plays a role in lactose synthesis that we all know. It is also having a molten globule state. What it does, it helps in the emulsification process. It is also having a calcium binding ability that we have already seen. Also, it helps in apoptosis in tumor cells. Now, the second term in the title was the nanotubes. Before understanding what nanotube is, let us understand what is nanoscale material. What are nanoscale materials? So there are various nanoscale materials right now that we have already seen. So this can be simple matrix, the core shell, the dispersion, combination, nanotube, solid lipid nanoparticle, nanofiber, nanocapsule, nanosphere, and whatnot. So what are this? This nano is derived from a Greek word for dwarfs. Okay, and this nanoscience is defined as a study of phenomena which is related to the materials which are at atomic, molecular, or macromolecular scales, where the properties differ from those at a larger scale. Okay, And what is this nanotechnology? Nanotechnology is a derivative of chemistry, physics, and microfabrication techniques. Nanotechnology is the manipulation of material at dimensions between 1 and 100 nanometer. And this is the nanotube, which here it is highlighted, which we are going to see further. Let us see further. These are the applications of nanoscience in food technology, food science and technology. These are the materials that I've already seen that are nanoparticles, nanoemulsions, nanocomposites, nanostructured materials, the structures that we have already seen. Okay. And this have application in processing the product. Obviously, you have seen the delivery purpose, the formulation purpose, the packaging purpose. Also, it has a very important role to play in food safety and biosecurity. That is nanosensors and nanotracers. And there are many more applications that we cannot enlist here. Now, there are two approaches to get these nanostructures. One is top-down approach and another is bottom-up approach. In the top-down approach, what we do, we break the bulk material into fragments and then further we break down into nanostructures. In bottom-up approach, we take smaller molecules and bring them to form a nanostructures. Okay, so this is the introduction part. Now let us see that what is alpha-lactalbumin nanotubes and how they are prepared. So first of all, the protein nanotube formation. So there are two methods by which we can obtain the protein nanotube formation. This is the protein structure, and there are two ways. One is chemical synthesis pathway, another is introduction to devices. The devices such as pulsed electric field, coaxial electrospinning, or microfluidics can be used for the formation of nanotubes. Okay, the other way is the chemical synthesis pathway, in which the proteins are broken down to several building blocks, and the bonds are further formed in between them to get the nanotube structures. So this chemical synthesis pathway is the way by which our alpha-lactalbumin nanotube structures are formed. So we will see this in further detailed way. So this is the procedure by which we get the nanotube structures. Obviously, this schematic diagrammatic pattern. So there are three major steps involved. First of all, the peptide or protein building blocks are formed from the intact protein. Then the cross linkers are provided by which the self assembly will take place between the building blocks. And the third is the bond formation by which we'll get the self assembled nanotube structures that we're able to see here at the end. Now, these three steps, we are going to see how they are taking place. First of all, the peptide or protein building blocks here, how we are getting this. So the protein that we have already discussed is the alpha-lactalbumin. By introducing the alpha-lactalbumin to proteolytic enzyme digestion, we'll get the peptides. And those peptides, we are going to identify that which peptides are formed for this nanotube formation. So we have done MS peptide mapping. So we will get a chromatogram like this. So here I have covered two research in which in one research, this chromatogram was obtained, and these were the fragments identified from this chromatogram. Okay, so these are the hydrolytic products after adding BLP into alpha-lactalbumin. We'll discuss what BLP is in further slides. So then we'll see that what are the peptides. Okay, the second chromatogram obtained was this, and these were the hydrolytic products that were obtained. Now, let us see what similarity was found in both of these studies. So these were the fragments that were common, which were found in both of these studies. Okay, now let us take this second table forward and see what are the actual fragments of the alpha-lactalbumin which is participating in the building block formation. So this is the primary sequence of amino acids of alpha-lactalbumin that I showed you in the initial slides. Okay, and these are further the fragments. This is the table 2 from the previous slide. And these are the fragments that are involved in the nanotube structure formation. And here are the spaces where actually the hydrolysis has taken place. I would like to highlight that A is the intact protein of alpha-lactalbumin. That means unhydrolyzed alpha-lactalbumin is also playing a role in nanotube formation. Okay, now what is this BLP that came in picture in the previous slide? the enzyme which is used for the alpha-lactalbumin hydrolysis. So this is an extracellular protease, which is this BLP is Bacillus licheniformis protease. Okay, and this is very much specific to the peptide bonds containing aspartic and glutamic acid at the carboxyl moiety. But if the BLP concentration is higher, then it can also hydrolyze other peptide bonds than aspartic and glutamic acid. It is having a its action depends on the pH also, and the most important point over here, it is having a GRAS status, so it can be used in food without any problem. It is acceptable. So let us see further. So these are the reasons because of which BLP is used for the enzymatic hydrolysis of the alpha lactalbumin. Now let us move forward to the second and third step. So that is the self-assembly of the building block and also the cross-linking and the elongation process. So this is the schematic diagram. Till this we have observed. Okay, this hydrolysis by BLP we have observed till this that monomers we have obtained these monomers. Now what we are applying the calcium for the formation of dimer. Then again in the presence of calcium the nucleation will take place. So this type of structure will be obtained. Then further the nanotube elongation will take place in the presence of calcium and we will get a helical structure like this. So this is the final nanotube structure with the outer diameter of 20 nanometer and inner diameter of 7 to 8 nanometer. This can also be presented in a schematic diagram like this. This is a helical structure. This is a better picture to understand. Okay. So this is the alpha-lactalbumin nanotube structure. Let us move forward. So now this nanotube formation has taken place. We'll see that what are the different factors which is affecting the structure of alpha-lactalbumin nanotubes, right? So first, let's see that what are these nanotube structures look like before moving forward to understand the factors affecting it. So this is the alpha-lactalbumin hydrolyzed by BLP. So no nanotube structures are observed here. Then this is the TEM of alpha-lactalbumin nanotubes. This is the SEM of alpha-lactalbumin nanotubes. You people might be able to find these nanotube structures in this pictures, right? This is the gel obtained after incubation. After the nanotube structure formation takes place, it forms a gel. Okay. Now this gel has been positively stained, and this is the image that we are able to observe. Further, this is the negative stained electron microscopy. Here, the clear picture of the nanotubes we are able to see with the helical in between them, with a hollow diameter in them. And this is the picture, high resolution image, just showing the right-handed helical structure that we already observed in the previous slides. Now, moving forward, some information about the alpha-lactalbumin nanotube. So what is special about it? This is the first man-made nanotube. It is formed by the hydrolyzed alpha-lactalbumin, which acts as nuclei for the formation of the nanotube. Also, as I mentioned earlier, unhydrolyzed alpha-lactalbumin is also involved in the structure of nanotube formation. Calcium is playing the role in nanotube elongation that we have mentioned earlier. The length of the nanotube may vary from 100 nanometer to few micrometers. And the hollow tube is having outer diameter of 20 nanometer and inner diameter of 8 nanometer. So because of this diameter, which is in the nano range, we call it as nanotube structures. It doesn't depend on the length. It depends on the diameter, whether we call it as nanostructure or not. Now, the tube wall thickness is 6 nanometer and the pitch is 110 nanometer, and it is a 10-start helical structure. The chemical bonds involved in the non-covalent, the chemical bonds involved in the nanotube formation are the hydrogen bonding, electrostatic interactions, hydrophobic bonds, van der Waals interaction, and van der Waals interactions. Now, this is the 10 images of various stages during self-assembly of alpha-lactalbumin into nanotubes. So first image is after few minutes after incubation with calcium. B is after 20 minutes, and at the end of the incubation, we are having the nanotube structures with us. So these are clear images of the nanotube structures, which are very clearly observed. We can observe here.